Okay, so let's continue with our discussion. We've already talked a little bit about the diversity of cells and also that they share some common features. So here I have another link to a video that I invite you to watch. This time it's looking at the goings on and the multiple activities that go on inside a typical cell. And again, these animations are based on many experiments and data that have been collected over many years that together give us a little bit of a picture of what goes on inside a cell at any moment. Okay, so back to our discussion of unity. Because cells all have the same basic chemistry inside, whether they're cells of bacteria, as you see here in this scanning electron micrograph of in part A of the figure, or butterflies, or plants like a rose, or mammals like dolphins. Inside of the cell, there are some very common mechanisms that occur, including the breakdown of food to produce energy and the consumption of that energy to build the molecules that make up the parts of the cell as well as the inner workings of the cell. So this similarity is really important in our understanding. Of course, as biologists, we want to understand the relationships between all living organisms. And this similar chemistry can help us to draw conclusions about the relatedness of the organisms that we know exist here on Earth today. So at the top of this figure, which is a tree of life, we see a lot of diverse organisms, from plants and animals to bacteria and photosynthetic bacteria. And here on the y-axis you see is represented its time. So we're talking many millions of years. So from what we understand of the relatedness of the organisms we can know and study today, we understand that there could have been an ancestral cell that's common to all the organisms today, because we see so many similarities in the cell's structure and chemistry of the cells and the composition of the molecules inside the cells, whether it's a plant cell or a bacterial cell. And in addition to that, there were two major events that evolutionary biologists propose occurred in order to make the progression from this ancestral cell that was most likely prokaryote in its cell structure to the eukaryotic organisms and their cells that we know of today. The first was the acquisition of mitochondria. So you know from your Bio 195 that mitochondria are the powerhouses of the cell. But one interesting feature of mitochondria is that they carry their own DNA. So in addition to the DNA found in the nuclei of plants, animals, and fungi, there's DNA in the mitochondria, and that DNA so the interesting thing about the so one of the events that occurred early on in the evolutionary history of eukaryotes is that there may have been an early eukaryotic cell or a pre-eukaryotic cell that was a cell eater and may have engulfed a prokaryotic cell and acquired it as part of its cell structure. So we know that one of the major differences between prokaryotic cells in organisms that we see here on Earth today and eukaryotic cells is that eukaryotic cells have a mitochondria. Prokaryotic cells do not. But one interesting thing about mitochondria is that they have their own ribosomes. So there are ribosomes inside the cytosol and on the ER of plants, animals, and fungal cells, but there are also ribosomes inside the mitochondria. And these ribosomes have the form that is similar to bacteria or prokaryotic cells. And the animals, fungi, and plants in their cytoplasm have a different form of ribosomes. So while they all of these organisms have ribosomes, we can tell the difference between a prokaryotic ribosome and a eukaryotic ribosome. In addition, more recently, in evolutionary history, the acquisition of chloroplasts by plants has occurred. And so we see that there's also a similarity between photosynthetic bacteria and plants in their chloroplasts. So it was thought that then some plant cells had acquired, or maybe a parasitic photosynthetic bacteria, or somehow acquired the photosynthetic bacteria, which are much older, as part of their cell structure, and that the acquisition of these tiny photosynthetic organisms by plants led to their having chloroplasts in their cells today, and the capability of carrying out photosynthesis, which is part of the definition of plants, and makes them unique from fungi, which don't carry out photosynthesis and lack chloroplasts. So this theory of acquisition of some early prokaryotic cell into early eukaryotic cell is called the endosymbiont theory, where some relationship of symbiosis, or the coexistence, and perhaps even the co-benefit, or the benefit of both organisms, allowed the mitochondria to persist as part of a eukaryotic cell. So what advantage did the mitochondria give to the eukaryotic cell? These early eukaryotic cells were then able to grow in the presence of oxygen. And at that time in evolutionary history, the atmosphere became rich in oxygen. So the timing of this event, where we see now eukaryotic cells that contain mitochondria that are able to grow and multiply in the presence of oxygen, coincides with the acquisition of, or the appearance of mitochondria. So these similarities between very ancient prokaryotic organisms and very diverse organisms that exist today as bacteria or archaea, with us as humans, and also even plants or other animals, non-human animals, or fungi with us as humans, is really key to understanding more about our own physiology, our own genetics, and our own cellular features. So cell biology of humans can be studied by using model organisms, and here are several examples. E. coli being an example of bacteria that's commonly used in the lab to understand genetics and cellular mechanisms. E. coli are very cheap, they're easy to grow, they grow quickly. Arabidopsis is a plant. It's actually a weed, grows readily in the laboratory, and can be used for many genetics and understanding molecular mechanisms that occur inside the cells. There's also mice that serve as good models for diseases, human diseases. So we have mice that are models for diabetes, for cancer, etc. Canorhabidis elegans is also a type of parasitic roundworm or nematode that has very specific, exactly 959 cells in it. And so we can understand development by looking at the development of this worm, multicellular development, because this is a very simple multicellular organism. On the unicellular level, we have yeast, which, like E. coli, are very simple and grow rapidly, but because they are eukaryotic, they're a form of fungi, so eukaryotic in their cell structure, their mechanisms and organization of the cell applies more closely to that of human cells. And then, of course, understanding genetics, which you learned a lot about in Bio 195, the fruit fly, or Drosophila melanogaster, is a great model for understanding genetics. So, bottom line, why are these model organisms useful? Because they reproduce rapidly. We can control and breed them in the lab. They're fairly cheap to grow and maintain, and we can grow many organisms and isolate macromolecules and understand the function of these macromolecules in the cell. So how do we know that there are really similarities between species? And how do we know that these similarities are really based on genetic similarities or functional similarities of the genes? Well, one example is from your book is here where a child has a loss of pigmentation on the forehead similar to in the mouse. And both of these cases, the KIT gene is involved with mutations in the KIT gene that led to the loss of pigmentation in the skin or in the fur. And this phenotype is called, so what we see as a result of the gene, the mutation in the gene is called a phenotype, and that's called piebaldism. So we've seen examples, but how do we really know that the similarities or the molecules that we find inside a human cell are similar to other organisms? So there's a classic example that's outlined in Chapter 1 of your book about the CDC2 protein encoded by the CDC2 gene. And CDC is the cell division cycle, C for cell, D for division, C for cycle, or for short we call it cell cycle. It's a protein that controls the progression of the cell cycle. So this gene, if it's defective, will not allow for the cells of the yeast, for example, to divide. So when comparing the CDC2 protein from humans and yeast, scientists were able to show homologous gene products. So if we sequence the gene found in the humans, which is on the top row here in these two lines, sequence the gene product or the protein, so find out the amino acid sequence of the human form of this protein, that's at the top line here. And then there were two different forms of yeast or single-celled fungi that were studied. One was Schizo cerevisiae pombe, and the other is Saccharomyces cerevisiae. In this diagram, you could see in green, these are areas where the amino acids are identical. So threonine, valine, leucine, etc. In the areas where they're white, they're not identical. Or they may be identical, for example, here between the two yeast species, but different in the human. Now, some of these differences are not very big differences, so leucine here in the yeast and isoleucine in the human. But still, they're differences. So maybe it's that these proteins, while they look similar in their sequence, don't actually function in the same way. So, how do we really know that they work in the same way, that the human CDC2 really works the same way as the yeast CDC2? Well, we can use genetic engineering to answer that question. So first, there's an assay where you can make temperature-sensitive mutants. That means that when the mutant is grown at the permissive temperature, the lower temperature, it grows fine. And so look at this one outlined by the dashes. When the same or replicate plate of yeast is grown at the higher temperature, that yeast clone does not grow. So there's a space here on the plate. Okay, so there's a mutation in the genetic makeup of those yeast that does not permit them to grow at the higher temperature. And that mutation can be induced by chemical mutagens in the lab. So how does this help us to understand the question of are these human proteins and the yeast proteins functionally similar? What you can do is genetically engineer the yeast cells to carry either the yeast, the DNA of yeast. So here we have the Schizo cerevisiae pombe yeast cells, and we could introduce DNA from the other type of yeast, the Saccharomyces cerevisiae. By introducing that foreign DNA, even though it's the same organism, it's a different genus and species of that type of organism, the yeast. So we're introducing basically foreign DNA into the S. pombe. And if we find that we can get growth at the non-permissive temperature, then we know that the S. pombe have acquired a functional form of the CDC2 gene. Similarly, we could take human DNA and genetically engineer the S. pombe yeast to acquire fragments of human DNA. Any of the S. pombe that pick up the functional CDC2 gene from the human DNA would then show growth at the non-permissive or the high temperature. Voila, the answer, so that test was done and the answer was yes, we could find functional mutants such that the replacement of the yeast CDC2 with the human CDC2 gene, that DNA, produced a functional CD2 protein that could act to help the yeast progress in the cell cycle. Okay, so yes, they're functionally homologous. This yeast with the mutant, its own form of the CDC2 not working, could then reproduce with a human form of the CDC2. Any questions? So many studies of genes, of human genes have been done in this manner, and we understand a lot more about what happens in human cells because of these models, such as the Saccharomyces cerevisiae. Okay, so size is important in cells. take a moment to think, why are cells so small? And what advantage does this small size give to cells? Well, really, it has to do with the nature of the reactions, what's going on in the cell. The cells carry out all of the metabolic reactions, or all of these chemistry, all of their metabolism is happening within your cells. And these chemical reactions are exergonic, or exothermic, and they release heat. So, what is the advantage of having a smaller size cell? Let's just do the math and look at the surface area to volume ratio. So 1 millimeter, okay, a cube has 6 sides. 1 millimeter times 1, so the length times the width times the height, would determine the volume of the cell if it was a 1 millimeter cube. So 1 times 1 times 1 equals 1. So 1 millimeter cubed is the volume, whereas the surface area is the area of each of those 6 sides added up. Okay, so 1 by 1 would be a 1 millimeter square times 6. So you have 6 millimeters squared. So 6 to 1 is the ratio of the surface area to the volume. So if we look at the larger cell, that's 20 millimeter cube, and we calculate the volume, it would be 20 by 20 by 20. That would be 8,000 millimeters cubed. And the surface area would be 20 by 20 is the area of one side, so 400 times the 6 sides, or 2,400. So that ratio ends up being less than 1, whereas the ratio of the smaller cell is 6. The amount of nutrients that can get in a cell at any one time and the amount of waste that can get out, or heat that can get released from the cells, is much more readily facilitated in a cell with a large surface area to volume ratio. So smaller cells are advantageous in this way. So how small exactly are our cells? Well, here's a great image from your book that shows going from basically the size of your thumb, which is a little more than 20 millimeters, all the way down to the cells in your skin, to the subcellular level, so the cells in your skin being about smaller than a millimeter. And if we look closely at a single cell, we're talking about somewhere around, if it's stretched out, maybe 10 microns, but human cells range from 5 to maybe 12 micrometers. Then the organelles within the cell, here's a mitochondrion, are on a much smaller scale. You're going down to 2 micrometers or less. And then the other organelles, such as the ribosomes, which are not membrane-bound organelles, but simply a combination of protein and RNA, are much smaller, on the order of nanometers. So here you can see there's a single ribosome is about 20 nanometers. And then we're going down to the atomic level, where the actual atoms in that protein subunit of the ribosome are on the order of 0.2 nanometers. So from the smallest units of the chemistry of life, the atoms, all the way up to the size of a cell, which can be as large as 200 micrometers. You should understand the relationships, kind of the general size or the general trend in sizes, going from nanometers to micrometers to millimeters, of the components within cells and the cells themselves. And you should memorize these conversions, okay? So 10 to the third millimeters equals 1 meter. You go down another log order to get to another 3 logs orders to get to micrometers. So 1,000 micrometers equaling a millimeter, and then again 1,000-fold down to nanometers. So 1,000 nanometers equaling 1 micrometer, or a million nanometers equaling 1 millimeter. These units of length, you should memorize both the units of length and the units of volume, which similarly have the relationship of 1,000-fold less. 1 milliliter is 1,000-fold more than a microliter, which is 1,000-fold more than a nanoliter. Okay, so I've told you a little bit about some molecular biology and genetic engineering that can be used to study cells, but really much of what we understand about cells is by looking at them. And so we can look at them under the light microscope, and by staining we can understand and distinguish different parts, including here the nuclei in pink with the basement membrane or the plasma membrane in blue. So in addition to staining, we can look under the microscope at live cells. So staining, you have to actually kill the cells to look at them and study them under the microscope, because the staining process itself will kill the cells. But if you want to look at live cells, you can also use light microscopy, just by using the diffraction of light to see the details within the cell. Still, we're limited by light microscopy because it only gives us a certain amount of magnification. The resolution is somewhere around 0.2 microns, and the magnification is about 1,000 times. So electron microscopy, which doesn't use light but uses a beam of electrons to examine the specimen, can give quite a lot more detail because of the shorter wavelength of the electron beam. So here you can see labeled in dark, this dark line here is the plasma membrane. You can also see the nuclear membrane, which is a lighter gray membrane, so the nucleus is up, kind of chopped off. You only see part of it here at the top. Right around that you can see the many layers of the endoplasmic reticulum, as well as these larger round structures that have multiple internal membranes, enfolded membranes of the mitochondrion, and compartments like peroxisome, as well as the lysosome, which has lytic enzymes which help to break down materials inside the cell or that are engulfed into this and brought into the cell. The peroxisome has important peroxides that, if not compartmentalized, could be detrimental to the functions and the chemical reactions that occur in the rest of the cell. And then we also have ribosomes, these tiny dots studded along the ER here, that you can view. You would never be able to see under a light microscope, but with electron microscopy they are brought into clarity. So electron microscope gives a much higher, especially the transmission electron microscope, which gives these transmission electron micrographs that you see here, and gives a lot more, not just magnification, but also resolution. So you can see, instead of the scale being up at 50 micrometers, we're down here at 2 micrometers, and we're looking at things that are about 2 nanometers in size. So we can see a lot more detail. I just want to remind you that you can go and see videos of cells online at the Garland Science website. and this is a really cool video of an amoeba. So this single-celled organism, eukaryotic in its cell form, can actually move by changing the organization of its plasma membrane, thanks to the cytoskeletal features. So now I'm going to just review what hopefully should sound very familiar because you've already learned it in Bio 195, and that's the structure of the eukaryotic cells. Okay, so eukaryotic cell is bound by a plasma membrane. It's a lipid bilayer. That plasma membrane contains the cytoplasm, which is everything including the organelles. Okay. The liquid portion that these organelles kind of float around in and are suspended in is called the cytosol. So that's everything but the organelles. So what's in the cytoplasm? In this liquid portion of the cell called the cytosol. The organelles that are included, usually the biggest one is the nucleus. It's certainly the most important as far as the brains of the cell, containing the recipes in the form of the genetic material. This is where DNA replication occurs. This is where transcription occurs. And so it's a key feature that, and also just from the name eukaryotic, as we already discussed, it's the definition of the eukaryotic cells that it has a nucleus bound by a membrane. The mitochondrion is also a membrane-bound organelle. It actually has two membranes. It has internal membrane system as well as this external one. And this is the powerhouse of the cell. It's where ATP is produced. So that really important energy form. That's the energy currency of all cells. Here is an image of the chloroplast. Let's just look at the one in the center. This electron micrograph, you can see the chloroplast has, again, multiple kind of pancake layers of membranes, lipid bilayers, that contain chlorophyll, as you can see in this colorized diagram of the image. The chlorophyll that's contained within these many layers of membrane-bound compartments is green. The rough ER. So there are two forms of endoplasmic reticulum. There's the rough ER and the smooth ER. The rough ER is basically the factory where many of the proteins that are destined for delivery to the plasma membrane, the outside, or to be secreted outside of the cell, are produced. The rest of the ribosomes are found in the cytoplasm, where the intercellular proteins are produced. So remember that the messenger RNA then exits the nucleus and finds ribosomes, where it's then translated to protein. The Golgi apparatus is the delivery system of the cell. It's many membrane-enclosed compartments in a very densely packed kind of pancake shape. And vesicles butt off from one end to the other. And as the proteins that are carried in these vesicles go through these compartments, or these many layers, they are modified post-translationally. So they may get glycosylated. Finally, there's also the endocytic vesicles and exocytic vesicles. So endocytosis, endo meaning in and cyto meaning cell, is the process by which the plasma membrane pinches in and brings materials from the outside of the cell to the inside of the cell. and exocytosis, exo meaning out and cyto meaning cell, is just the opposite, where membranes pinch off from internal organelles such as the Golgi if this is a molecule that needs to get outside of the cell, be secreted from the cell, or get to the plasma membrane and remain there as part of the membrane. It would pinch off the Golgi apparatus, and that vesicle would travel and fuse with the plasma membrane in the process of exocytosis. The cytoskeletal components of actin, microtubules, or intermediate filaments, actin being the very smallest, microtubules being the very largest, are all involved in keeping the shape of the cell. They're also can be readily disassembled or reassembled. They help to keep the organization not only of the organelles within the cell, but also help the cell to move in the case of motile cells. So all together now, you should know all of these internal organelles and all of the terminology here on this slide, including some external features which are seen in eukaryotic cells such as cilia. Any questions? Okay, so now it's the time for you to get to work and memorize or re-memorize from your days of Bio 195 the organelles as well as their functions, and review Chapter 2, the kind of basic biochemistry, for next week. So here are the objectives that I expect you to know from Chapter 1. And here are some practice problems that you should be able to do. Okay, so just a reminder that online you can always do the chapter post quizzes. So this is after you've read and taken notes. Close your book and take Chapter 1 and Chapter 2 quiz. Go back over the answers and go back to any areas where you where you made mistakes and review that material before moving on. And then there are a couple questions from your text that would also be helpful to know in preparation for the exam. And you can look up the answers in the back of the book to find that material. Again, I would suggest after you've done the reading, after you've taken notes in class, and just to close your book and try to do these without looking at your notes.